This is SR823. It should be on the screen here. So HC2479. This is this sample over here, but you see that it doesn't show up on the map because there is no location for it. Okay. Maybe what we can do is we can look up where is the city of Jacmel. It's over there, and we can look up and fill in some data that is missing. Okay. But the other thing that we can do here is this. We can color the dots by any of the columns. Okay? So for instance, we've got this column called source, and we can say it's I'm just trying to, maybe it's this, but it's this one right here. We can choose which color should we use. At the moment it's using the site column to color, okay? But I could color by source, okay? I'm going to switch this to be relating to the SRA. And if we look at the phylogeny done like this, then what do we see about our samples and their relatedness? If we look at this, okay, now what I haven't, if we look here, here's the metadata on date. So if we look if we look at 253, right? The 253 that we were discussing earlier, right? 253 is over there at the top, just below the reference, okay? And remember that 253 was quite similar to 373. When we looked at the SNP distance matrix, we saw that 253 and 373, they're quite similar, right? So we see that there. Then they are collected, 373 is here, collected in 2015. More than 2 years later, after 253, okay? Then 370 is collected in 2014. So somewhere in the middle time-wise. But 253 is collected from the environment. Okay, let's zoom in again on the map. You see the environmental collection is here along this river. Okay? And 373 and 370 are collected from clinical samples. Then, and if we look at it in time, the order goes, the 253 is before any of these other ones, 373, 370. But then later on, more divergent, remember these were more divergent from the reference, 364 and 260, okay? So 260, as we can see from the color, is an environmental sample. 364 is a clinical sample, okay? And 260 is collected in 2015, 364 in 2017, okay? What this means is that after being introduced by a human traveling in a plane from Nepal, is the current theory, into Haiti, this pathogen has established itself in the river systems, in the water systems of Haiti, and the human cases are clustering together with the environmental cases. There isn't environment on this side and human on that side. What we're seeing is a movement of pathogen from human to environment and back again. The environment has established itself as a reservoir for pathogenic cholera in Haiti. Okay, because we know that cholera is not an obligate human pathogen, It's not like Mycobacterium tuberculosis that you're finding pretty much only in humans. It's well, I think in great apes as well, but it is something that can easily go hide in the environment. And having done so, of course, it's much, much, much more difficult to eradicate now that it has found its environmental niche. And it's the same thing now, SARS-CoV-2, even if we were somehow able to get rid of SARS-CoV-2 in every human, it's in cats, it's in deer, it's in so many other things, you know? So yeah. So our approach to cholera in Haiti is certainly needs to be lots of wash interventions. I don't know, what are you going to do about the fact that you've got pathogenic cholera in your river? I mean, can you get rid of the cholera in a river? Is it possible? You can control, but the thing is that now you must never let your guard down, because what happened is that they managed to stop the initial outbreak 2010, but it keeps coming back. Now, most recently in 2020, they've had another, outbreak. So you have to stop you've now got a thing that is no longer coming from outside, some travel or whatever it is, but it's really embedded in your community. Okay. So that's microreact. I'm not going to go deep into it, but you can do something very similar with next strain. If you want to use next strain, you use this. You can install it locally. Microreact, not so easy to install locally. Next strain you can install quite easily locally, but you can also use their web interface from Auspice. I think it's meant to be pronounced auspicious or something. I don't know. The auspicious website. Same kind of way of working, that you can drag a tree file onto it. And there's the tree. And then you can drag a okay, so this one, it couldn't figure out how to link the two things together. I think that's because the sample name was not just the SRA name. But so auspicious, it works slightly differently, but it's another similar tool. I probably just need to restructure my metadata to use it. Okay, so have other people managed to get this picture on their screens? Okay, now let me look at all the people who've not managed to get that picture with the broken metadata and things like that. I want to show you one more thing, but let's see what your metadata looks like. You just get to a terminal. So let's go back to the web browser. I wonder if it's a problem of I have never worked with this thing. So we can definitely, if we go upload, we can definitely make it work with just a tree, right? So if we take, okay. I don't think the order matters, but just in case. No? And it's going to spin in circles again. Oh, does the order matter? Okay, let's go back and try and reproduce that experiment. So if we go on metadata, and now we go tree. Okay, that's it. We just discovered that if you are going to do metadata with microreact, you must put your tree first and then your metadata. If you put your metadata first and then your tree, it just spins in circles. So you have to do tree first, then metadata. Okay? and then it does work. Where's the metadata empty? Which empty metadata?
On Auspice, it's meant to process data from another group called organ.
Yes, but it can also just do it.
Yeah, it do it, but the organ means profit divided something. And Auspice is a token, it's a divided token. So that's the basis of the name.
But maybe we'll have some time. Maybe tomorrow I can show tree time, because then when you run tree time, what you get out is a Auspice format JSON file, and then you just drag that thing onto the onto Auspice and it displays. Both of them are amazing systems. Both are amazing systems, but I must admit, with Micro React, I don't know why it's not showing here, but you can actually make it do a timeline. Like, I just can't remember how to do that now, but I think it might be because my dates. Maybe you can do that.
But we don't have a tree file, do we?
A what?
The snip, it's pilot tree, it's not a time tree.
No, it's not a timesheet, but what I'm saying is that you can, there should be a view down here at the bottom that has cases over time.
Oh, I see.
But I'll have to look at it. The Micro React you can actually use for more than phylogenetics, because one of the things, but I've shown you an example with Micro React where where there's no data in the metadata, but you can actually load metadata into the into Micro React where there's nothing in the tree, and so you can use Micro React to visualize your cases. If you've just got a line list with cases, you can visualize the cases. You can make it basically draw an epi curve. So it really is quite a powerful tool. But if this it's its own thing of learning. Anyway, so who else has got trees or not trees? Okay, so let's see what you've got. Okay, so let's see what happened with this. Did you get it? Did you get your Micro React working? I haven't checked my front row yet. But your thing was having issues. Your computer was having lots of issues. But you got it working, yes? Oui. Well, yeah. Your computer has issues like Ivan's computer. Okay? But did you manage to make it work? How? Okay, so we should upgrade her, in other words. Can you sit with Ivan and upgrade your computer? Okay? Okay. The paper was about the change in serotype of Ogawa to Inaba, the antigenic shift. Okay? So my understanding of the biology is not perfect here, but if I understand the papers correctly, there is some theory that the switch on methylation from methylated to unmethylated on the polysaccharide outside of the Vibrio cell wall is related to the growing immunity in the population. Okay, so this maybe people are better with cholera biology than me can verify that that's true. But how this comes about is by a change in a gene. Okay, so in the paper that one of the papers that I use as a base for this work it states. Sorry, do you want some help just getting is your microreactor working before you move on to the next thing? Is it working? That is looking beautiful, but you can just put the labels on using this button over here. Yeah. Then you go to loads of labels, and then you go to leaf labels, switch it on. Boom. And then you've got labels, and then you can choose which is your label and things like that. Okay. So the serotype that was introduced into Haiti in 2010 was Vibrio cholerae 01 mTOR Ogawa. Okay? And then later on, they discovered that there were lots of Inaba strains around. Okay? And the difference seemed to be related to a change in the WBET gene. Okay, so this led me investigating this led me down a real rabbit hole. Okay, because if you go look in the literature for WBET, you very often don't find it. You find that it's also called RFBT, okay? And they've changed the name of the gene over time. Then if you look in the annotation of the files that we use as a reference here, they have neither a reference to WBET, nor do they have a reference to RFBT, okay? So it's like, what gene is this thing? Luckily, on VicPred, I was able to find the nucleotide sequence of the gene, okay? And using BLAST, I was able to find it in the reference genome, and using that, I was able to find what they call this darn thing in this particular annotation. So this is the problem that we have. Data is not laid out on a plate for you sometimes. You have to go hunt for it. So the end result of this was that, let's just see which window I have this in. Oh, wait a minute, it wasn't my other working directory. I'll find my way around this again, just a second. You don't need to do this. I'm just doing this to remind myself of the right name again quickly. Subject. Oh, I need the FASTA file of this one. Let's go. Let's see about it. There's the hits. So it's a reverse strand thing, so it starts here, and not phosphate GenBank. Okay, so this gene is known as this in this in this GenBank file. Okay, but what next? Okay, so if we look in the Bactopia SNP folders and we choose one of the samples like 373 or 364, and then we look at the SNP, and then we see that there is a something or other dot annotated.vcf.gz, okay? So now I'm going to take this, and what I want to know is, in all of my files, do I see any mutations? Sorry, I need to go zcat, all of these things. And then I need to just put a star here, and the star here, and pipe integrate. And we see that in a few places, there are mutations in this gene, okay? And specifically what it is saying here is that in this gene, in these samples, there's a frame shift variant. So what's happening here is that there is a GC. If you look over there, the GC gets turned into a G, which leads to a frame shift, which leads to a truncated form of the protein, which is exactly the marker for that shift from Ogawa to Inaba, okay? So I'm just going to unzip, just for myself, just so that it makes my life easier. I'm going to unzip all of these VCF.gz's so that I can now do my grep a little bit differently. It's going to give the same result at first. Now I'm going to say grep minus L to see which files I find it in. So in these two files, 260 and 364, okay, I see a shift. So using that information, I'm going to change the metadata.csv, and I'm going to add another column, and I'm going to call this thing serotype, okay, and reference, sorry, I need a bunch of commas here. That's the SOA, comma, for nothing in the publication, comma, and this one was Ogawa, and the next one was Ogawa, and then if we're just looking for 260 and 364. So 260, there's 364, that's comma, that's Inaba, this one. What I'm doing is I'm annotating I'm manually based on my grep, but I could automate this with a bit of extra effort. I'm annotating the serotype of the sample. So we need one more over there, and this is a Garwin.
Is this for microreactor?
For microreactor, so we can have a look at it. Okay. And now I'm going to go back to my microreact. I hope that I haven't messed up my format of. I'm going to go back to my microreact. I don't know if I can actually just drag straight onto this thing. Let me see what happens if I drag. drag this onto here. Can I replace the metadata? See what happens. No, I have to restart the project. Okay. Department, this one. Department. Okay, I'm just going to load this file into LibreOffice just to make sure that I haven't totally messed it up. Which I probably have.
While Peter's busy with that, you understand what's happening. Yes? No?
So I was trying to very manually add this column. Okay, it looks like I didn't do too bad a job because there it is. I should automate this, never manually do stuff like this, because manually doing stuff is how you make mistakes. I'm going to abandon this project and start again. Sorry, so where's my file browser? First I need my tree, and then I need my metadata. Continue. Did I break something? I must have broken something. I'm not quite sure what I've broken, but I'm going to have to go back and look at that thing. But what I wanted to show you is that you could then use, we can do this tomorrow, but you could then use MicroReact to color things based on the biotype. And what you'd see is that from the snippy that we've done, the prediction of the variant in that gene, which we know is the RFBT, otherwise known as the WBE gene, we can actually predict the biotype of Vibrio cholerae that is in our sample. And what you'll see if you look at this thing relative to leaving aside MicroReact, if you just look at this in LibreOffice, what we notice when we look at the dates of the Ogawas and the Inabas, we notice that the Inaba samples are collected in 2015 and 2017. Here, towards the end of 2015, and then 2017, and the Ogawas are collected at earlier dates. Okay, so what we see is that over time, as the infection persists in the Haitian ecosystem and continually reinfects the population, we have what appears to be selective pressure towards a serotype switch from Ogawa to Inaba, okay? Which is the kind of thing that we expect to see with organisms when they're under selective pressure in environment. We expect to see something happening, some mutation that happens by whatever chance become then fixed in the environment. But yeah, sorry, I don't know why the MicroReact doesn't like my file. I'll figure that out overnight. But yeah, putting it all together, okay? I haven't shown you the other way of typing, where we use MLST and all of those kind of things, which is also something that Bactopia can do. I just don't have time for that. I will see if I can get into that a little bit tomorrow, okay? But putting it all together, my point with this was that with Bactopia, and all the other tools, like you see, we've used what George taught you about Conda and Docker, okay? We've used Nextflow. We've used understanding how to work on Bash, okay? And now we've used Bactopia. Put all of these things together and you start having a toolkit of abilities to work with bacterial genomic data and metadata, visualizing using microreacts and so on and so forth. The specifics of how you work with your particular organism, whether it's Mycobacterium tuberculosis or whether it's Salmonella typhi or whatever you happen to be working on, or whether it's a virus, are obviously going to change depending on the biology and the research questions. But I hope that you've seen that just using these tools, putting it together, and your laptops, okay, which we've managed to tweak some really nice work out of. Almost everybody's laptop is performing splendidly, that putting this all together, then we actually have a bacterial, a pathogen bioinformatics toolkit. And that then based on this and building on this, and hopefully the network that we create between ourselves, what we've been discussing, Tony gave us some ideas, we had some discussions last Friday, we can start seeing that what other pathogen projects are possible, especially if we tackle them collectively between us. And I think that's enough for today. I think there is afternoon coffee. So I can help with any problems and configurations and things like that people need. And then tomorrow I want to see if I can work with a few other techniques, which was a bit more experimental. I know that one of the things I haven't spoken about much is the nanopore data. Part of the reason is that it's such a wild west and the techniques are not as well polished. But yeah. Okay, coffee time and question time and all other things.